So the panel is being led by Dr. Petra Kaufman, who is the director of our Office of Clinical Research. She's been here about 3 years. Oh my God. 3 years and a few days. We managed to recruit her from Columbia University, where she was a tenured professor, and she has transformed our clinical research programs. Petra, it's all yours.
Thank you very much. So we are running a little late, so I'll start right ahead. And I also understand from the organizers that we be given for this session an extra 10 minutes because there's a lot on this agenda for this hour. So we won't quite have a full hour, but a little longer. So I'm really pleased to introduce the panelists today here. I start with Dr. Ed Kay on my left, who brings a lot of very relevant experience to this session, not only as a clinician, as an academic researcher, and then more recently, maybe not that recent at this point, in industry where he, as you probably know, has a really amazing success record in terms of bringing a drug, helping bring a drug with for a very rare disease, Pompe's disease, to regulatory approval. He's currently the vice president and chief medical officer of Sarepta, a company that also deals with rare diseases, and he can maybe add to this if I missed some important point. And then the other panelist is Dr. Amelie Gubitz from the NINDS here. She's a neuroscientist. Prior to joining the NINDS, she was working as a researcher at the University of Pennsylvania on spinal muscular atrophy and in the laboratory. And since being at the NINDS, she has worked on several diseases, currently on ALS in the neurodegeneration cluster, and she brings a lot of experience in bridging between different partners. So what we thought we would do is we really want this to be interactive. And as you see, this session has two parts. So we'll have the first, I guess, 20 minutes at this point. And the topic is building resources to be ready. So what do we all need to be ready to bring a new treatment to a neurological disease from the industry, from the NINDS perspective, and from your perspective. So we really want to have your questions. So get ready to ask what is on your mind in terms of readiness for trials and setting the stage for success. And each of the panelists will just have a couple of slides, I think, to share with you their thoughts and to open up the session. So Dr. Kay.
Well, it's really nice to be in a room full of people who are very interested in the brain and the central nervous system. And even as a young student, I was always fascinated by the brain. But then I had the insight one day, and I said, you know, the organ that I think is the most important in the body is the organ that's telling me it's the most important. So I was receiving biased information, but I still think it's a good idea. But one of the questions that people will ask me, and well, how do you have a company? And in my now I'm working in a biotechnology company and had worked at Genzyme for 10 years, and now still staying in the rare disease area. But what is a company interested in? Why would they work on your disease? And what I can tell you is that most companies are agnostic. They don't really care what disease they work in. The thing they care about is what is the risk. And if you as a organization can show them that you can reduce the risk and you have a greater likelihood of success, then they will become interested in your disease. So what is it that, and again, and many times we've, as a company, you decide, what are the areas that you want to focus on? And a lot of it is we will bring in experts, certainly in the disease, but also the patient organizations, and they have a huge influence on what we'll study in. And so one of the things that the industry is interested in is what's the population? How many people with this disease? How many could be amenable to therapy? The other important aspect is what's the natural history. If you're going to design a clinical trial, you have to know what the natural history is, when patients are likely to get sick, how long does it take, what can you expect for a therapy. We also need to know what the medical experts are so we can work with them. And a very important aspect is, do you really understand the biology or the pathophysiology of the disease? So as an example, in our company, we have avoided diseases in which we don't understand the biology. We've tried to focus on diseases such as Duchenne's muscular dystrophy, where you have a protein, there's a deficiency in the gene, you don't have the protein, and the hope is that if you can replace the protein, you should make them better. So that's the kind of biology that we like to understand, because then you can develop biomarkers to see if your therapy is actually working. So again, critical things. Is there a regulatory precedent? What I mean by that is, has somebody gone before you, and does the FDA understand the process, and are there clinical endpoints that you can use? So that reduces the risk. Understanding, for instance, as an example, in Duchenne's, the 6-minute walk test has now become very important, and in data that we've just reported, the 6-minute walk was in fact the best test we had. So it's turning out then the FDA is likely to take that as an endpoint. Biomarkers are really important, and one of the things we've been looking at very intently is to actually measure dystrophin, which is the protein that's missing in Duchenne's muscular dystrophy. And if you can replace it, that's a good biomarker. And then also really understanding the natural history. But I guess the important aspect, well, how do foundations assist in this? And one of it is, we've heard really a registry of patients, first of all, identifying patients where they live, understanding the natural history. Also importantly is understanding what is the standard of care, because in any trial, you need to make sure that the patients are currently receiving the best care available, because if they're all receiving different levels of care, you can never have a homogeneous population, so that on top of the standard of care, you put your therapy and you see a difference. And I think that's something that we're very interested in. Also, informing the patient community about trials. Many of the patient groups that I've worked with, the ALS, the Prize for Life, SMA Foundation, certainly all of the Duchenne muscular dystrophy groups, PPMD and Cure Duchenne and the MDA. What they have done is they have a very savvy group of patients who know about their disease. They understand the genetics of the disease. They understand what research is being done, and they're committed to helping a clinical trial. As an example, we worked, when I was at Genzyme, Petra mentioned, we worked at Myozyme, which was the first therapy for a neuromuscular disease, for glycogen storage disease. That trial of 90 patients was enrolled within about 3 months, and it's because the patients knew about it, they wanted to participate, and even when the therapy was approved, this was for the late onset population, they could have gone out of the trial, dropped out, but they stayed in the trial. We only lost 5 patients out of 90. And the reason they gave is that we know that this is important to have continued approval of your product, so we will participate, even if that means for me that I may have to go, in this case, it was 18 months without therapy, because if they were in the placebo group, they weren't getting therapy. So that means that they really understood the importance of research. They understood the regulatory aspects of it and what it meant. And that was the foundations that informed the patients of what needed to be done. And part of it also is that I think from the advocacy groups is really informing the FDA about your disease, because the FDA doesn't know about the 6,000 genetic diseases or 7,000 genetic diseases. They can't possibly know. So the more that you can inform the FDA about the importance of your disease, the natural history, why it should receive accelerated approval. And there's been some really interesting changes in the law. The PDUFA 5, which is the FDA Funding Act, has really put in rare diseases as a focus, as accelerated approval. So in fact, it has lowered the bar a little bit to get a disease out there. And I can give you some examples, one of which is Vertex Pharmaceutical, that had funding from the Cystic Fibrosis Foundation. Well, their product was approved in 3 months. Remember, by law, the FDA has to approve it in 6 months. That's accelerated. Typically, they have a year. Well, they approved that product in 3 months. Why? Because it was important. It was a life-saving drug for the community. And a lot of that, I think, goes back to the CF Foundation. And even a company like Pfizer, which is the largest pharmaceutical company in the world, they're doing research in CF that's solely really funded by the CF Foundation. So what are they doing? They're throwing their money at a project that's really important, and they're accelerating the process of the disease. So I think it's one of those things I think that really is very, very, very critical. And I think, finally, the question is, well, how much is important? Well, what can you do? Is money important? Of course, you hate to say it, but money is always important. There's no way to get around it. But I remember an example of when I was at the University of Pennsylvania, at the Children's Hospital, we had made a diagnosis of a rare genetic disease, methylmalonic aciduria. Very, very rare. We didn't even have had an animal model to work on it. And the father asked me, what can I do to help? He was a single individual. And I said, well, we don't have an animal model to work on. He goes, okay. So he goes back to his local, the state of Pennsylvania representative, and got a line item in the budget for $50,000. And he came back to me in the clinic. He goes, "What can you do with $50,000?" And I said, "Well, let me think about it." And $50,000 seems like a lot of money, but it really isn't for research. It's a small amount of money. But I had one of my fellows who was Chuck Venditti, who's now a senior investigator at the NIH, and I said, "Chuck, can you make for $50,000? Do you think we can make an animal model?" He goes, "I'd be willing to work on it for a year." And so we got money from the University of Pennsylvania, from the Department of Genetics, hobbled together a group, and within a year we had an animal model. Well, Chuck then took that animal model to the NIH, and now he's doing gene therapy in this methylmalonic aciduria. So that $50,000 turned a very bright young investigator onto that disease. That's the kind of change I think an advocacy group can make, because you're getting people, and you want the young, bright people interested in your disease. Chuck couldn't care less what disease. He just wanted a genetic disease, and he wanted to make a difference. And because of that father, he has, and that disease has progressed. So I'll end here.
So before we open the topic for discussion, Petra and I just wanted to very briefly show you 3 more slides to give you a little bit more information about NINDS. And one point that we wanted to re-emphasize is that NINDS is currently engaged in multiple initiatives that were specifically developed to build resources and infrastructure for clinical trials and clinical research. So one of these programs is our brand new Neuronext Clinical Trials Network. Dr. Landis already talked about this network earlier today, so I don't have to give you additional detail, but the only thing that we wanted to emphasize again is that this network is open to researchers from academia, from foundations, and also from industry. So we encourage these researchers to propose their trial ideas to NINDS to be conducted in the Neuronext network, and these trial ideas and proposals are then being evaluated by peer review and also by our advisory council. So in addition, NINDS has also developed a range of tools that have the goal to enhance clinical research. So one of these tools are the NINDS Common Data Elements. These are content standards that have the goal to harmonize the collection of data in clinical research so that this type of data can more easily be shared amongst investigators, and it's also more readily available for meta-analysis. So far, NINDS has developed one set of common data element standards that are general and applicable to any disease, as well as over 10 sets of common data element standards that focus on a specific disease. And all of these sets of common data elements are freely available on the internet. So any researchers can utilize these standards in his or her research in order to advance his research faster. And another tool that we have supported is the NeuroQual assessment tool. This is a patient-reported outcomes tool to measure quality of life in neurological clinical research. And again, the goal is to provide an off-the-shelf tool that is accessible to researchers to incorporate in their research. And finally, we wanted to emphasize that NINDS takes a very holistic view when it comes to therapy development. Of course, we recognize that there are many stakeholders who are engaged in this very complex and important enterprise, and really no one can do this alone. So certainly foundations and patient advocacy groups are a very important player here. We understand that many of you are engaged in a broad range of activities, but for the purpose of this session, we really wanted to focus on clinical trial readiness, especially since you have the patient knowledge and also understand the patient perspective. So Ed already discussed many of these resources. I think the important take-home message is that these resources are building blocks for therapy development. They should be thought about in the process very early, and they can certainly help you attract private sector investment to your disease, since they will de-risk therapy development. So I think I'll end here, and we'll be happy to take any questions, and I'll let Petra moderate.
Would you please come up to the mic or get a mic and perhaps tell us briefly who you are and yeah, and what you exactly, where you...
Megan O'Boyle with Phelan-McDermid Syndrome Foundation, a patient advocacy group for a rare genetic syndrome causing autism and epilepsy. We have a patient registry. We've just completed our first year, and we're revamping it to make it shorter for longitudinal data. Is the last tool that you mentioned something that we could include in our registry? Is it copyrighted? In other words, if we could use those set of questions and have our families answer them, and then the data would be available to all researchers.
So I think that maybe were you talking about the NeuroQual?
Yes.
And yes. So both the NeuroQual and the common data elements are publicly available for free. They're not copyrighted, so you could use both of them. And there's advantages. So there's currently common data elements. There are some general ones that you could incorporate into your registry, and the advantage would then be that these are used already by other groups, so that potentially, there could be some advantage in the future for putting data together. And the NeuroQual also is available.
Great,
so the websites were, I think, on this. You have the slides, I think, right? Mm-hmm.
Hi, I'm Michelle Rhee from the National Brain Tumor Society. Not that Michelle Rhee, different Michelle Rhee. I had a question about the CDEs and the NeuroQuals. Are they sort of validated or approved by the FDA, so the FDA is willing to use them as sort of secondary endpoints or whatever the case may be?
So regarding the common data elements, there's currently no standard that is approved by the FDA, but we are working with the FDA in one disease. For example, in Parkinson's disease, we have collaborated with CDISC to have the common data elements for Parkinson's disease also mapped to the C-DASH standard. That's a standard that is used by industry and also by for submission to the FDA. So we are in contact with the FDA, but since there is currently no single standard that is approved, nor are the common data elements. Regarding NeuroQual, again, the FDA qualifies outcome measures in the context of use, so not in sort of a general context, but there is an interagency committee on which both Dr. Moy, who's the program officer for NeuroQual, and myself serve, where we're trying with SEALD, that's the division in the Office of Drugs that works on the qualification of outcome measures, to see if we can achieve that. But it will always be in the context of use. It will not be a blanket approval, unfortunately. That'd be nice. Any other questions?
Oh, yeah, we have plenty of time. We're going to run this session for 10 more minutes. Okay.
So this is Robert McBurney from the Accelerated Cure Project for Multiple Sclerosis. I think that a lot of patient groups are interested, or lots of patients are interested in, so of the existing therapies, what's the best therapy for me? And that's, of course, the area of personalized medicine, or precision medicine, as it seems to be redefined as of this weekend. So a lot of what you talk about is clinical trials for the development of new therapies. It seems to me that the area of running trials that collect biomarker, longitudinal outcomes trials that collect clinical and molecular data, or the samples to generate molecular data to develop predictive tests to support personalized medicine, which in a way is a low-hanging fruit in health economics, but also a very important area for patients. And in clinical trials, there's always the responders and the non-responders, and working out what it is that makes someone a responder and someone a non-responder to something that seems like a very good therapy is excellent. So what's the role of the NIH in this activity, and can the same NeuroNext, for example, be used in the context of those sorts of studies? Because I think that it's something that many of the pharma companies are not going to do, because they're primarily interested in their treatments being with patients, and so that needs to be work in that area.
Don't want to answer all the questions, but they seem to be directed at me. So yes, so certainly we are interested in both. For most of our diseases, there is not that luxury that we have a lot of treatments and we can figure out which one is the best for whom specifically. But where that is a problem, certainly that's something we would also be interested in. So from our perspective, NeuroNext, for example, could very well also look, if that was an important question, at a subpopulation and see what treatment is best for that population. But I wonder if maybe there's an industry perspective on that question of sort of, I don't know, do you have anything to add?
Yeah, no, I think one of the things that if you're working in a disease area, not necessarily a specific therapy. So an example is been involved with approvals on 3 different drugs over 10 years. But one of the things that we would have to do first is really get together a group of experts and also patient groups and identify what are the standards of care. And a lot of it has to do with, what is the physical therapy that's necessary? What does that really bring? What is the pulmonary care? If you take a disease that has multi-systems, that's really critical, because if you don't apply that standard, you will have very different outcomes. And so you need to standardize the care. So one of the things that happens sometimes in working and concentrating in a rare disease, there are several publications that come out, and then there's an educational process that the care actually for the majority of patients, and typically globally, gets better because they're getting ready for a disease and they're really trying to improve the outcome. And I think that's the first step. And I could see something like NeuroNext being able to do that, because you could compare different standard of care measures, and you could see what is really working. And I'm also I'm also on the board for the NeuroNext project for the SMA biomarkers, and that you're right, that would never be done by industry. It's too expensive and it would take too long. But that is something that if biomarkers come out of that work, every company will use that, because they can take that biomarker. So I think this kind of collaboration with NeuroNext makes a lot of sense.
One last question.
Just to follow up on that previous question, I mean, this issue of stratification, obviously it's very costly and it differs from disease to disease, but even just guidance, even just sort of principles to be thinking about when attempting a stratification type exercise, like the questions that you brought up, responders versus non-responders, or how do we even go about approaching some of those questions? I mean, certainly, so Press for Life is developing a really large database, and we're hoping to look at issues of stratification and issues of slow versus rapid progression in ALS, but there's not really any process out there. And I wonder if that's the level that NINDS could engage in, so more at a guidance level, as opposed to, obviously we don't expect you guys to compile all the data from every single trial that's been, or to get at these levels of personalized medicine or predictive medicine, but that level of guidance might be an area where NINDS could really take an active role.
Right. So, as you know, the FDA is the agency that would issue guidance for investigators planning their projects. But I think that
just at the scientific level, I meant.
At the
just
scientific
at the question,
level, yes. Yeah. So certainly. You already made the point that basically, if there are large data sets as the ones that you're compiling now, then you can go into strata more easily. So in phase 2, which is where NeuroNext is located, as you know, we try to have a specific population and try to choose the one where you can have some success in the proof of concept and recognize a signal. But then clearly later on, if one wanted to stratify, large data sets would be required. And I think what we are looking into now is how can we get these relatively large data sets. One thing is by combining data from different trials or specimens. So for example, things like the common data elements or data archiving could then help if all, let's say, ALS data for clinical trials or registries were collected using elements that can map to each other, then you would eventually position the field in much better to do these kinds of important analyses. And another area we're interested in is also how we can use data, for example, with the increasing use, hopefully, of electronic medical records. Is there something that we can do other than running very, very large clinical trials, which by definition, given our budget and the fact that we have 700 diseases to attend to, we cannot run very large trials for all diseases. And many diseases, there are not enough patients for these very large trials that would allow stratification. Thank you very much.
Anyone else want to add anything?
Thank you
Very much.